Welcome back. Today, I would like to just quickly revisit the topic of lipids. Right, so we've talked about lipids a couple different times in the context of all of the biomacromolecules. Right, but lipids are a category of thing that we as living organisms make and require to stay alive. Right? And so the other ones are carbohydrates, proteins, and nucleic acids. But we're going to ignore those ones today. We're only going to care about lipids. Right? So we're going to focus on these lipids. And lipids are technically any biomacromolecule that is soluble in non-polar solvents. Right? So if I put this in a non-polar solvent, this thing is going to dissolve. That's what that means to be a lipid. Okay? And what that really presents as in a more useful light is that these are fats, oils, waxes, and steroids. Those are the categories of stuff that we engage in this world that are lipids. Okay? The fats are lipids that are made up of these fatty acid chains. Okay? And so fatty acid chains are these long carbon-based chains with hydrogens sticking out most of the parts, right? And on the very front, we have a couple oxygen-based groups. Okay? This is one fatty acid. To make a fat, we combine a bunch of different fatty acids and build them up into one molecule. Now, if we're dealing with a solid fat, most of those fatty acids are going to be these saturated fatty acids that you see here. It's a straight line because they're all single bonds. Yeah. The point of these fats is primarily to store energy. Right? Each of these bonds required a little bit of energy input to build, and so if I break those bonds, I get some energy out. So these are effectively energy storage mechanisms. Right? You can see a lot of bonds, a lot of energy being stored. The reason why the solid fats are mostly saturated fats is that these saturated fats can stack neatly on each other and they build up a solid mass. Right? So you get this nice tight stack, you get this nice solidy fat. Right? If you think about fat, like it's not the most solid thing in the world, but it's solid, it will stay together. Right? You warm it up and it'll start to melt and break apart. But for the most part, these are solids. Okay? Oils are pretty much the same thing as fats, except for they're liquids. Right? Same function, same base monomers, same idea going on here. The main difference is that they are primarily unsaturated fatty acids, right? So they are the ones with these kinks in them. Right? You get this one little double bond here, it makes this little bend. Okay? And because they have these bends, they don't fit together very nicely. Okay? And so you get a lot of space between each of these little fatty acids. It makes them more loosey-goosey. It makes them liquids at room temperature. But other than that, fats and oils are pretty much the same. They have the same function, energy storage. Then we have waxes. Right? And waxes are really simple single chain fatty acids. So instead of stacking a bunch of fatty acids together to make a fat or an oil, we just have this one chain and it's just chilling. Yeah? And so these individual chains are able to interact with one another very easily and they kind of glob up. Right? And so if you think about waxes, you get that glob. Right? If you really pull out a good piece of airwax and you stare at it, you'll see that it has like little individual filaments almost, like these little pots that jut out. And you can see that it's just a big amalgamation of these individual strands. Right? So you get these jagged edges. They stick together, right? But you can mush it because they're all the individual pots. Right? And that's what a wax is. Now, these waxes function primarily as waterproofing layers and as antibacterial layers. Right? So if you think about your ear, you don't want water going into your head and you don't want bacteria going into your head. Right? So these wax layers help stop those things from going in and out. That's the primary function of wax in living organisms. And then we have steroids. Steroids have an entirely different shape from other lipids, right? And so the other ones are all based in fatty acids. The steroids are based in these ring structures. So these are the monomers of steroids. They got these big fancy carbon rings. They're still dissolvable in non-polar solvents. Right? And so that's the important part here. The primary function of steroids is for cellular communication in our body, right? So if I need to send a message from my pituitary gland down to my liver, right, I can send a hormone from here, it goes in my blood, gets to my liver, and sends the message. Okay? Now, because these are not soluble in water, they don't dissolve as they flow through our blood, which is very important. We don't want our signal molecules to fall apart along the way, right? And so these hormones can persist for a very long time in our bloodstream because they are nonpolar. If they were polar, they would dissolve in our water-based blood and just fall apart. We don't want that. And primarily the reason we're revisiting lipids right now is to explore their role in the membrane. And so the fact that lipids are nonpolar makes them hydrophobic, right? And so they don't interact with water. This is very important when we consider the role of lipids in our membrane, right? You have to remember that like we are primarily water, right? So outside of our cell is water, inside of our cell is water. There's a whole bunch of watery stuff going on in our body. So if I want to keep my individual rooms separate, I need barriers that don't let the water stuff go through very easily, right? And that's what I'm going to use to separate my rooms, my cells from one another. And so the lipids provide this hydrophobic layer, right? And so it keeps my watery stuff on one side away from my watery stuff on the other side. Now, the most simple demonstration of this is in these single-layered lipid balls called micelles. Right? And so these are literally just, you got one layer of lipid in a circle, in a sphere. Right? And so the outside stuff is watery, and the inside stuff is anti-watery. Right? The entire stuff is going to be non-polar stuff. Maybe it's a ball of fat inside. Right? It's a ball of oil inside. Okay? It's something not water-based. And so this sphere of lipids keeps whatever's inside safe from the water outside because the water can't go through this non-polar grouping. Okay? As we extend that out into what we actually use for our membranes, right, we have a double-layered membrane setup. Right? And so the inside is watery and the outside is watery. So I need two layers because my inside of my membrane needs to be non-polar, but the inside, this region here, has to be polar to be comfortable with the watery stuff inside. Okay? So I need my polar regions to be comfortable with my water areas. Okay? And then I need my non-polar regions to keep my water areas separate from one another. Okay? So these are basically like the walls in your house. Right? Like you can't pass through walls, and that's why walls work. If you could pass through walls, walls wouldn't do walls' job, right? And so it's due to the non-polar nature of lipids that we can actually have these membranes, and we can have distinct little areas within our cells that let us specialize in the functions that we need to, so that we can be the cool, awesome, living things that we are. Right? So that was our quick revisit to lipids. Hopefully it got you either some new information or refreshed you some information and helped you understand why we care about lipids' non-polar nature when talking about things like cell transport. That's it. Have a great day. Bye.